Good morning. Welcome everybody on our joint seminar for SAME-NeuroID initiative within Horizon Europe Twinning program. When I say this, I always look at Witek Konopka from some reasons, and our department, of course. It's my great pleasure to welcome today and introduce our speaker, Aleksandra Badura, is from Erasmus Medical Center in Rotterdam, which is one of our partner institute in SAME-NeuroID program. Okay. Aleksandra has done her master at Jagiellonian University in Krakow, and then she moved to Erasmus University when she did her PhD, defending in 2011. And then she moved for a postdoc to Princeton University, working with Samuel Wong on cerebellum. And then she did another postdoc also in Netherlands. Is that correct?
Yes.
Okay, cool. I did not mess up. And in 2018, she established her own laboratory at Erasmus Medical Center, where her research is primarily focused on cerebellum function and learning in the pathological and physiological states. So Aleksandra is using experimental tools, experimental approaches, combining them with modeling, and she managed to develop some program which allowed to predict motor dysfunction based on neuronal activity in the cerebellum. So as you know, cerebellum is well known for coordination, controlling coordination of movements, but it does a lot more, and Aleksandra probably will be telling about her other line.
No spoilers. No spoilers, please.
No spoilers. Okay. I will finish very soon. So her other work is on association between cerebellar dysfunction and autism spectrum disorder. Her work and other demonstrated that in autism there is frequently a malfunction of cerebellar neuronal activity, and that trauma during birth of cerebellum insult can be associated with development of autism later on in the future. What's worth adding that Aleksandra has developed some tools to study in the automated way complex behavior, which is particularly of interest for Michał Ślęzak and myself. Aleksandra, the floor is yours. Let's give a little applause to welcome Aleksandra. Thank you.
Thank you so much. Thank you for really warm welcome. And as I was saying, no spoilers, because I always like to ask the audience, what do you think the cerebellum does? And then I hear a lot about sequences, motor function, etc. And here I will be talking mostly about the non-motor functions of the cerebellum. I also adjusted the talk because at the Neurones conference in Krakow in a few days, I will be talking only about new ways of quantifying behavior. So if you have questions about these automated ways, non-supervised ways of quantifying behavior, please ask me after the lecture, because today I will be talking to you about the role of hind and midbrain inhibition in autism-like behaviors. So my lab is really interested in cracking this puzzle of heterogeneity in autism spectrum disorder. We know it's a spectrum, and maybe some of you have heard this quote: if you've met one person with autism, you've met one person with autism, meaning that really everyone is different. This is something that everyone's very unique. Neurodiversity is also a theme here. And that, of course, makes diagnosis very challenging. But yet, when you talk to people who are on a spectrum, very often you can notice that they have some common traits. And those are the common traits that are independent of the etiology of autism. So here I wanted to show you two pictures. In the middle, this is Robert, who's sitting, and Tom, his father. Robert is a NF1 patient, so neurofibromatosis type 1, which very often also is comorbid with autism. He is diagnosed with autism, but is at intellectual norm, and he actually works at Erasmus MC as a biosafety officer. He knows all the chemicals, so it's like a walking book of all the chemicals that we use at Erasmus. And here on the right, this is actually my brother-in-law, Jeroen, who's also diagnosed with autism, but his etiology is hypoxia at birth, and he also has a mild intellectual disability. So both of these men are completely different. For some of you that are maybe football fans, one is for the Rotterdam team and the other one is for the Ajax team. When it comes to football, it cannot be more different in the Netherlands. But they have those common traits. And when you talk to them, you do notice that they share some communication deficits, particularly in following intent, following social discourse, and also some of the, maybe not necessarily repetitive behaviors, but more stereotyped type of behaviors. So the question that I and others want to answer is, what are these neuronal origins? What are the common shared neural mechanisms for these shared phenotypic traits, given that no matter the etiology, we can observe the same outcome. And indeed, when I started my postdoc in 2012, cerebellum was really not on the map when it comes to autism and the role of the cerebellum in autism. So we wrote this review where we looked at all the clinical evidence, and the clinical evidence was fascinating because it said that cerebellar injury at or around birth, particularly in this third trimester and around birth, carries the risk for developing autism later in life that is only comparable to having an autistic identical twin. So it's a major risk factor, and the question was why? And why is it also around birth? If you get a damage, or I would get a damage to a cerebellum right now, I would most probably not develop autistic traits. I would be probably quite ataxic though. So you will see all these motor coordination deficits. So what is it so special during the development? So this is the some of the evidence that we already knew from the clinic. It's what was shown with people, with autistic people post-mortem, was loss of Purkinje cells, and in MRI pictures we could also very often see hypoplasia of the vermis. This is the central part of the cerebellum, and also in general very often a smaller volume. We also know that the cerebellum is not only connected to the areas that are relevant for motor output and motor coordination, but there are extensive connections, reciprocal connections with the forebrain regions. And there's a lot of work that's been done on it. We've also done some tracing experiments ourselves, where we show that both in, if you look at fMRI functional connectivity in humans, or tracing experiments in non-human primates like rhesus monkeys, and in mice, there are these connections with the forebrain, with MPFC, and also with posterior parietal cortex. So I will go through this quite quickly because this is all published work and I really want to show you the unpublished data. But we embarked on this journey of understanding what happens when you silence parts of the cerebellum, when you disturb the activity in parts of the cerebellum, either throughout the development or acutely when the animal is an adult. And we use dreads. Are people here familiar with the dread technology? For those of you, I see some people, but probably all of you have heard of optogenetics. So where you can, with light, activate a neuron or silence a neuron. So dreads are very similar. With viral injections, you deliver a receptor, and this receptor can then bind to a ligand. When it binds to a ligand, it again activates and silences the neuronal activity. It does it for a long period of time. So whereas with the optogenetics, when the light is on, it's on. When it's off, it's off. Here you give a ligand, and then for several hours the activity is disrupted. And this is really nice because you can give this drug in drinking water, and by giving this drug in drinking water, you can disrupt the activity for weeks or months, and then you take this out from the drinking water, and within a few hours, this drug is going to be taken out of the system. So we disrupted the activity at postnatal day 21, and then continued throughout the development, or we only disrupted the activity acutely in adult mice just while doing the behaviors. And we employed several very classical ways of measuring mouse autism-like phenotype. I always like to say autism-like because, of course, it's not one-on-one of the behavior in humans. So this is this very classical social chamber where the mice can hang out with a novel mouse or with a novel object, and mice, as being social mammals, they like to hang out with the novel mouse. And what we saw is that when we disrupted the activity of CRUS1, those are all controls, and you can see that there's this preference of spending more time with a mouse, with a novel mouse. But when we disrupted CRUS1 developmentally, or CRUS2, the mice didn't have this preference. So they would spend equal amount of time with a novel mouse as with a novel object. And this was important because we only saw this phenotype when the disruption happened developmentally, but not when it happened acutely in an adult mouse. We also saw quite a drastic perseveration phenotype. And there's actually been a paper just published in December last year that corroborated the studies using a different type of dreads. And they also showed, so I'm always very happy when someone replicates your study, you're like, yes, it works in a different lab. And this perseveration is quite dramatic. As you can see, this mouse just keeps on going. So it learned that the platform was here. Now we switch the platform to the other side. And even if we close off this arm, the mouse is just going to keep on swimming back and forth. And it's almost like not seeing the other arm. And we know that it knows that there is the other arm because in the beginning we do a habituation session where the mice can explore all the arms, and we make sure that they do swim in the very beginning in the habituation session to all the arms. But once they learn and once they consolidate it, it's very hard for them to switch. And this is something that we actually see in autistic people quite a lot. This inability to adapt to changes in the environment rather than learning. And as I said, this is case for reproducibility. I should put one more figure here in the 2023, but you could see that there's already been a paper in 2017, my paper, and another paper in 2018 following my paper that either used DREAD technology to show that there's this disruption and that there's this loss of the selection for the mouse. So here we have the selection from the object to the mouse, and here we have the loss of this selection. Or if you delete autism risk gene specifically in a cerebellum alone. So TSC1, I will tell you a little bit more about it in a moment. TSC1 is a tuberous sclerosis 1 gene, and it's an autism risk gene. So this is something that we and others are now working within this hypothesis that just like Hubel and Wiesel, when they showed that if you suture a cat's eye during development, the visual cortex cannot develop normally. And then if you open the eye after the critical period, these neocortical areas will never develop normally and the cat will never have a normal vision. And now this is how we know that there's a critical period in development where input shapes the maturation of the neocortex. We think that this is sort of the same, in the same way, the cerebellum during the development shapes the maturation of the relevant neocortical areas. Now, we're not saying that it means that the cerebellum is the social hub or anything like that. We are saying that this is, think about it more of like a GPU of a computer, right? It's parallel processing and it's thinking of, oh, something happened. This is an association that I'm saying, and I'm giving this information, this relevant information to the cortex. So today I want to talk to you about published and unpublished work that we do about the genetic insult to the cerebellum, but also to cerebellum and the midbrain, and how we think that that affects the autism-like behaviors. And this is work that is completely spearheaded. All credit goes to a senior postdoc in my lab, Dr. Ines Serra, and also Dr. Fabian Kaiser, who is an immunologist, and also he was a PhD student in my lab and now is a doctor, and he still collaborates with us. So we will be talking about the role of TSC and PAX5 in autism pathogenesis. So TSC1 is an important protein in the mTOR pathway. It forms a complex with the TSC2, TSC1 and TSC2. It sits right here and it inhibits Reb that then can activate mTOR. So mutations within TSC1 and TSC2 lead to a disease which is called tuberous sclerosis complex. It is a rare disorder, and it is it has a very complex multi-organ manifestation, because mTOR is important for cell growth, cell division, cellular processes everywhere in the body. So you have dermatologic, renal, pulmonary problems, but you also have neurological findings. And around 90% of TSC patients exhibit TSC-associated neuropsychiatric disorders. There's a whole big list of the neuropsychiatric disorders that are associated with it. A lot of them can be connected to tuberi in the brain. So if you do an MRI, you can sometimes see that there's abnormal tissue in the brain, sort of like a non-malignant tumors that are growing. And that, of course, would translate to psychiatric deficits. But interestingly, there's also quite a high population, and we cannot draw a correlation between having these tuberi and being diagnosed with autism. So there is nothing that can predict this is the person who's going to also have an autism diagnosis because they have these changes in MRI. And in fact, between 30 to 60%, depending on epidemiological studies, of people with TSC are diagnosed with autism. We also like to work with this gene because, as I showed you before, if you delete it specifically only in cerebellum, only in the Purkinje cells, the mice are already going to develop social deficits and also perseveration. And we also know that in a healthy brain, actually the expression of TSC1 and TSC2 is enriched in the cerebellum compared to other brain regions. So we wanted to find out what actually is happening in the cerebella of the TSC1 mice mutants, and we used a global mutation. So this, again, to be as close to the patient, we use a haploinsufficient loss of function mouse mutant, and then we looked at the cerebellum. So first of all, what we could see is that if you look at the wild type, those are immunocytochemistry stainings of a wild type and a haploinsufficient mouse at embryonic day 18, postnatal day 1 and postnatal day 7, we can see that there's an increase of phosphorylated S6, and this is a marker for mTOR activity. When mTOR is overactive, you're going to have more phosphorylation of an S6 protein, and this is like a readout that we know that this mTOR is pathological and has a pathological function. What we also found by doing a lot of qPCR is we found that there was an interesting loss actually of the PAX2 expression, and we confirmed it both with the Western blots, but actually also here when we quantified the PAX2, we saw that there was less expression using immunocytochemistry, fluorescent immunocytochemistry. And why is PAX2 really important? Because it's been already known that PAX2 is a transcription factor, and that transcription factor is critical for developing the inhibitory cells in the cerebellum. So PAX2 is the one that basically determines the lineage consequence during the development for the neuron, that this neuron is now going to be GABAergic neuron, and it's going to be an interneuron in the cerebellum. And indeed, when we state for parvalbumin is a marker for inhibitory cells in the cerebellum. Also in neocortex, cerebellum doesn't have somatostatin. For those of you who are into inhibitory cells, in the neocortex we have somatostatin positive and parvalbumin positive. In cerebellum it's all parvalbumin. What we saw is that there's much less PV cells in a cerebellum. This is a sagittal slice through a mouse cerebellum. You can see this beautiful folia. And then when we zoomed in and we quantified the parvalbumin, we saw that at P7, there's a deficit. We continued with this work because the previous work was published all the way up to P7. Now we continue with this work and we see that actually all the way to the P18, there is still a delay. If you quantify the interneurons in an adult mouse, you actually don't see any issues. So in an adult mouse, the interneurons are already all in place. But all the way to postnatal day 18, where a lot of pruning is happening, of course, a lot of maturation is going on in the mouse, we still see that there is a deficit in the expression of the PAX2, and this is both to the anterior and the posterior lobe of the cerebellum. This is important because the anterior lobe is more thought as having more of the motor functions, whereas the posterior lobe is connected more to the non-motor areas. So it's always really important to quantify different parts of the cerebellum, just like in the neocortex. And moreover, we also now can see that this is both types of interneurons, so-called molecular layer interneurons, but also big Golgi cells that are affected by this mutation, and they have this arrest in the development. So what does it mean? We also started to quantify the pup behavior because we wanted to see, okay, we know what happens in an adult, but what happens in a pup? And all credit goes to Ines, who introduced all these tests in my laboratory. It's quite difficult to work with pups. We start to work with them when they're P3, and they have a few of the reflexes that you can measure at this time. Working with pups at this age is very challenging because you actually have to introduce yourself to the mother, because if you just take away the pups and you do stuff with them and then you put them back, the mother gets stressed and it's very highly likely to actually eat the litter. So we already start to handle the mom when she's pregnant, so she knows the experimentator, that she's comfortable with the person who's later going to be doing the experiments. And there are also some tricks to it. So for those of you who would like to work with very young mice, please, we can discuss some of the tricks to how to work with them. And then we follow their development from P3 all the way to P14, because past P14 you can start to examine sort of more normal adult behavior, because at this point they already walk, they can see, they can smell, etc. And we didn't see any gross motor deficits in general. But when we looked at their social behavior, so this is a P12 mouse, and this is a tiny maze where we can test a social smell and a non-social smell. So what we did here is like a miniature Y maze, and you can have an almond extract and here a home bedding, and the mice are going to recognize these smells. What we saw, and this is an ongoing project right now, we were quite excited about this data, is that actually these mice have more preference to their own cage smell. So from the non-social, but also to stranger social, they really gravitate more than the wild-type mice. The wild-type mice are more likely to explore other smells, but they really want to have this type of sameness. So the sameness and the sort of safety of their own cage. And when we combined all these scores, we actually saw that when it comes to the motor progression, there is a non-significant, but yet if you look at the Cohen's d, the distributions non-overlap. Effect size in the juvenile mice when it comes to motor score, and a significant social progression score that actually then normalizes in the adult. So there's something quite interesting there, and we are wondering what is it during the development and what exactly happens when these interneurons are arrested, and what are the outputs at this age from the cerebellum when it comes to electrophysiology. So what is disrupted in the activity of the cerebellum when you also disrupt the activity of the interneurons. And this is why, the reason why we do it is because we would love to find some predictive markers. Something that could say, okay, if you have this mutation, but also we see this and those changes to the maturation of the cerebellum, then you are more likely to develop these types of deficits. There is a missing step though. And one of the missing steps is that we actually do not know how TSC1 and TSC2 complex can directly influence PAX2. So there is no direct interaction between TSC1 and PAX2, but there's been some actually really interesting work from Drosophila and also from some mouse embryos. So now we are investigating this protein UNC, and UNC is in the pathway where it can be influenced by TSC1 and TSC2, and also that can then regulate the PAX2 expression. But of course, this is now, our hypothesis. So now we actually want to go and show whether the UNC is also dysregulated in the TSC1 model, and show that this is the one that that leads to the dysregulation of the PAX2. So this is where we are right now with this project, and we're also started doing electrophysiology at those different developmental time ages. So we take the mouse, the brain, the cerebella of the mouse at P7, at P14, at P21, and at P56, and then we record the electrical activity. So I hope that by the end of the year, I'll be able to share that full data set with you and see how this arrest in the interneuron development leads to changes in the activity of the output neurons, so the neurons that then communicate with the rest of the brain. But one of the also interesting story is that if you look at the PAX2 gene alone, it regulates the interneuron development, but it also interacts with other genes that are important for the development of hindbrain and midbrain. One of them is NGRAIL2. NGRAIL2, Alex Joyner has done a remarkable work, her lab, and also Roy Silito in the US. They've done a remarkable work of really showing that those are the genes that are responsible for the foliation of the cerebellum and the development of the cerebellum. But whether there was a lot of work done on NGRAIL2, but not that much work done on PAX5 until in 2019, when this guy, this immunologist, PhD from Department of Immunology, who never did anything with neuro, showed up in my office, and he was like, "Hi, you're Aleksandra, right? So I'm Fabian Kaiser." He's very German also, and he was like, "I have this very interesting patient, and we made a mouse, and the mouse is weird." And I was like, "Tell me more. I love weird mice. This is my business, weird mouse stuff." So they found this patient mutation. This is the only patient in the world so far that has a biallelic mutation in a PAX5 gene. There are some patients identified with the haploinsufficiency, so when one allele has a loss of function mutation. We think that complete loss of PAX5 is embryonically lethal in humans. We know that because if you make a mouse model, they die around weaning age, the complete null knockout. This patient inherited one point mutation from the mother. This is the point mutation. And then had a de novo mutation that basically resulted in a truncation of this whole allele. So you can think about this as a null allele. And this point mutation, at the time when Fabian contacted me, they already did a lot of work in the immune cells, because this patient, next to having low IQ, hyperactivity, and autism spectrum disorder diagnosis, he also had severe immunodeficiency. So they first modeled the PAX5 function in the immune cells, in the B cell development, and then they came to me because of this neurophenotype. But there, what they show is that this one allele still produces the PAX5 protein, but this protein doesn't bind to the DNA as efficiently as the wild type protein. So this is PAX5 is also a transcription factor, right? It needs to bind to do its job. And this one is like binds and then falls off, and then binds and then falls off because of this point mutation. So by the time when he got to me, and then I started to dig in what is known about PAX5 in the brain, there was literally well, there were two papers. One was from Urbanek et al., and then another one was from Alex Joyner lab. And they made the haploinsufficient plus-minus mice and a global complete knockout. And they saw actually that the neocortex and the forebrain were fine, but you can already appreciate this decreased mass of the cerebellum. And they also showed that it's highly, highly expressed very early in the embryonic development in the hind-midbrain barrier. So the this so-called isthmic organizer, for those of you who are in developmental neurology, this isthmic organizer is full of PAX5 very early in development. And there's also been some findings from GWAS studies and clinical epidemiological cohorts that showed that haploinsufficiency, so again, losing one allele, increase the risk for autism. And those were also some incidental findings in large cohorts where this gene would pop up for risk of schizophrenia or major depression. So definitely something going on with the central nervous system. And then the other paper from the Alex Joyner lab that showed that this PAX5 interacts directly with the NGRAIL1-2. And again, NGRAIL1-2 is the one that is very important for cerebellar development, there's been a lot of work done. There's also been studies that looked, of course, at single-cell RNA-seq, and there's a lot of databases nowadays where you can look into it, and we know that indeed PAX5 is also critical for GABAergic lineage when it comes to maturation of the neurons. And this has been actually both now, there are beautiful data sets from the developing mouse brain, but also from the developing human brain. And there the PAX5 is linked to the developing interneurons, GABAergic cells. So we did some tests in the patient. This is a test for eye movements and hand-eye coordination, because we really tried to understand the sensory motor deficit of the patient, because he, if you interact with him, he doesn't seem to have, he's not ataxic, he doesn't have any gross motor deficits. But then when you challenge him, particularly with so-called anti-saccades or anti-tapping, what does it mean? Normally, saccadic eye movements is if I flash a target here, your eyes are going to make a saccadic movement to the target. That's very standard. But now if I ask you, fix your gaze here, ignore this target, and then when the target disappears, make an eye movement to the opposite direction, that's very hard. And that's very hard because it requires inhibition. It requires an inhibition of the reflex of your eyes to go directly to something new that pops in your vision. And the same for pro and anti-tapping. You have this urge to tap immediately to where you see it, and then you have to restrain this urge and to go to the other direction. We knew that the patient understood the task because his eye movements would make this small saccade to the target, and then he would correct himself, be like, had this really, you know, intense face of staring, and then try to go to the other side. But he was unable to suppress this reflex movements. And he was also pretty bad with memory saccades or memory tapping. And all of the latencies of the movements were longer. He's also extremely hyperactive. So it's impossible for him to sit still. He always fidgets, moves around. And then these are actually mice that we made. So we made the haploinsufficient mice and also the patient mutation, the biallelic mice. And in any test we would put it in, so in open field, in a grooming task, in a maze, they would always move more, always. Actually, we even called a few of these mice Max Verstappen, because this is a Formula One reference for those of you who don't know. It's like, anyway, because they would be so fast, for example, traversing from one side to the other, that it would really seem like they were always sort of running. It's also very difficult when you try to do analysis and be blinded, because if you stare at the cage for long enough, you could sort of be like, that's a mutant, that's a mutant. Not because of the weight or the appearance, but because they move a lot and they rear and do all these things. And then we saw an interesting phenotype in the social chamber. So again, this is where the mouse can choose between a novel mouse and a cup, or an old mouse and a new novel mouse. So this is the stranger 1 versus stranger 2, which is a social novelty. And what we saw is that actually these mice were hypersocial. And then you could tell me, well, they're not autistic, right? They're not autistic, they're not, they are hypersocial, and they should be hyposocial. But I think it's also sort of misunderstanding of an autistic phenotype that's been portrayed in the media, where mostly men, because if you think of media and how autism is portrayed, it's mostly men who are exceptionally brilliant, like Sherlock Holmes, Sheldon Cooper, whatever, right? The good doctor, who do not want to interact. But for example, both the patient and my brother-in-law, when they talk to you, they would stand like this, which I think makes you a little bit uncomfortable at this point, right? The patient immediately asks me and Fabian whether we're dating. In the first time, he was like, oh, this is your boyfriend, right? And they're like, no, I'm my PhD student. No, please not. It can be that it's not just a social, as we call it, social deficit, a lack of interaction, but the inappropriateness of social interaction actually is also something that is a hallmark of autism, and something that a lot of autistic people struggle with, because they don't feel like, where is this distance? Where is this social bubble line that we feel comfortable with? So it was interesting to us that these mice displayed this phenotype. By the way, this is also a phenotype that can be seen in like Angelman syndrome mice or Williams syndrome mice, where they are hypersocial. We also did sensory motor tests in the mice. We did rotorode, so this is where the mice have to walk on the rod and then they fall off. It's a very crude test, and with this very crude test, we could only see that it was the patient mutation that had a deficit. We also did eye blink conditioning. Eye blink conditioning is a classical Pavlovian type of conditioning where you pair an inert stimulus, like here, light, to an unconditioned stimulus, which is a puff. So we have a CS and a puff. And what happens that in the beginning, the mouse is just going to blink to the puff, to the unconditioned stimulus. But over time, it will develop this conditioned response, and it will close the eye to the light alone. This is a super conserved behavior. We can do it in humans. I'll show you in a moment why. We're very excited about the fact that you can do it in humans. But mice have this reflex, ferrets, cats. It's been also very, very well described. And we understand the role of the cerebellum down to the cellular level, like which cell do you manipulate, which part of the behavior it's going to disrupt, because it's been studied since the 60s, this paradigm. So when we looked at this, we could see that if you go from the first trial to the 200th trial, and this is the CR in the red. There's a CR amplitude. You can see that this conditioned response develops over time, and then at some point stabilizes. And in the haploinsufficient mice, even though they have a milder phenotype, I think you can immediately appreciate with those heat maps is that it develops later and is not as strong as in the wild-type mice. And then in the patient mutation mouse, it's sort of all over the place. They learn a little bit. So again, you know that there is some learning, but the learning is delayed. It's also very inconsistent. They have a lot of these weird blinks that they showed throughout learning. The reason why this is interesting to us is because we had to invite this patient to come to the lab. It was also COVID time, so it was very challenging because these saccadic eye movements, pro-tapping, you need a screen, you need like a nice eye movement device, a whole program, so you cannot really bring this to the patient. But with the eye blink conditioning, so this is, if you sum this up and you sum the amount of learning over days, you can see this difference between the wild-type mice, haploinsufficient, and the plus-minus mice. But the reason why we're very excited about this is because in a consortium grant that we got last year, we are working together with Blink Lab company. And this is the company where some of the, actually both my postdoctoral supervisor, Sam Wong, as well as a colleague of mine from Erasmus and C are involved in this company. They designed an app, and this app can actually measure this type of reflexes, the eye blink conditioning in humans remotely. And they just had the first paper published where they describe it, over 1,000 participants, but they also had measured autistic kids and kids with ADHD. And it's really nice because, again, you do it remotely on the app. How does it work? I want to show you a little bit. So this is, I'm going to need, because I'm going to also play you a sound, and I need this to play. So this is what you can see. This is a neurotypical child. They are watching a movie on the iPhone, and there's a camera that's also looking at their facial expressions. And what happens is that they have the headphones and they hear this sound. Did you hear it? Yes. And this is a sound that evokes a blink. If you have it on your headphones, it will evoke a blink. And then what we also can do is we can play a double sound, a small sound and a larger sound in between. And this is a classical so-called purpose inhibition. If you have a stimuli and then you have just before a stronger stimuli, it's actually going to cancel each other out. A lot of people in the doctor's office know that. So for example, if you need to get an injection, you can also pinch yourself somewhere else, and then the injection is going to hurt a bit less. So purpose inhibition works really well. And this is an autistic child. I should say that indeed all these images were used with the permission of the caregivers. And this autistic child also has a response to the, sorry, I'll play it again. Also has a response to a normal pulse. But now look what happens when you try to do purpose inhibition. There is no inhibition. Not only there is no inhibition, there's actually like almost an overexcitation with this pulse. And this is just a pulse inhibition, but we can also do a whole eye blink conditioning paradigm with this app. Why? Oh, sorry. I wanted to show you. So what they did is they looked at the controls, they looked at autism, and they also measured ADHD kids. And what is quite interesting is if you compared the ADHD to the autistic population, they actually can also differentiate between the two. So we're very excited about it, and we're going to deploy this app to the Netherlands Autism Register that has 1,000 participants, for which we also have genetic data. Of course, we're going to have our patient also play with it. And there's also been 16 other patients from 12 families that are PAX5 haploinsufficient that have been identified in North America with our collaborators. So we're also going to, we actually are receiving MRI scans from them, and we're also going to see whether we can see the common traits using this app. And why MRI? And this is also way sometimes we always say like, oh, you don't need more resolution and fancier machines. But here is the story of why fancier machines sometimes are important. So we had, this patient had a scan, neurological scan at 3 Tesla MRI at age 15. And the neurologists or radiologists say there are no abnormalities in the brain of the patient. But we were like, well, we would like to really make a very thorough analysis and really look at subcortical and cerebellar regions, which usually are not the parts that are the main focus. So we collaborated with someone who has a 7 Tesla MRI in Amsterdam. And we did manage to get the really nice MRI images from the patient, both from the subcortical regions, but also the cerebellum. It's very challenging because, again, he's autistic and hyperactive. So it took 3 sessions. First session, I went into the MRI to show him that nothing's wrong. The second session was the MRI was on, but he would just lay in and out. And then the third session, we actually played his favorite movie in the MRI. So he would lay still for the 19 minutes that it took to take a good scan, Lion King. So now we always now do this when we ask the people who are neurodiverse, is there a movie that you really like? And then you can play, because 7 Tesla is a little bit claustrophobic. Anyway, I digress. So what we found is that there was a hypoplasia of VTA and substantia nigra in the patient. And that actually reflected the MRI finding that we found in the mouse. So we were super lucky. There was an 11.5 Tesla in Vienna, and they scanned our mice brains. So you could really have a really nice resolution. You can see all the nuclei, all the folia in the cerebellum, subcortical structures in the mouse. And we found the same phenotype. There's less volume in substantia nigra and less volume in VTA. And also what we showed is that the total volume of the cerebellum was the same, but some lobules were smaller and some lobules were larger. So when you looked at the total lobe, and all lobules were there, but there was both hypo and hyperplasia of some of the lobules of the cerebellum in both the mice and the patient. So why do we think this is it? Oh, I don't know if you can see it, but of course in the mouse now we can do stainings. So here in the white dots you can see PAX5 expression in the SN and in VTA. In the haploinsufficient mouse, it's much less. And then in the patient mouse, there's basically no PAX5, almost no PAX5 expression. And then by doing a lot of co-stainings, we realized that PAX5 in these regions co-expresses or is only expressed in GABAergic cells. So the cells that express, this is an RNA scope, and you can see PAX5 in white. This is an NUN DNA, RNA, and here the GAD1 too. So we knew that it's expressed only in GABAergic cells. And then when we quantified those GABAergic only cells, we also saw this gradual decrease in GABAergic cell in the midbrain and in the VTA. And this is in an adult mouse, by the way. We have not done it yet during development. But in adult mouse, you can see that there's a decreased volume, and this decreased volume corresponds to having less inhibition. And we thought, well, VTA is super important for it's the dopaminergic output, right, for all the reward system. And then the SN is the dopaminergic output for all the motor system. So what happens if you don't have enough inhibition? Well, you might just be hyperactive and hypersocial. That's a theory. Well, that's a hypothesis where we're going. And as I said, we're now really excited that we have a collaboration with authors of this paper. So actually with Joel and the last author, Daryl Scott. Daryl, we from Harvard, we collaborate with them and we are getting MRIs of their 16 haploinsufficient patient to see whether they also show this hypoplasia. And what we have been doing in the last year is we've been doing some electrophysiology on the mouse brains. So we think, okay, if you have less inhibition, you will have more dopaminergic output. So the dopaminergic neuron is going to be overly excited. It's a little bit of a tedious work because you patch blind. So you have a mouse slices in electrophysiology, you slice the brain and then you patch a cell, but then you have to know which cell you're patching. And in the midbrain, it's really impossible to say. Like in the cortex, it's quite nice in the hippocampus to patch pyramidal cells. You can identify them. But in the midbrain, you have to patch and then stain. So we confirmed that that we patched the TH cells. And this is preliminary data. We're patching more cells right now, but we can already see that almost all cells that we have patched are hyper excitable, meaning that if you inject current into them, they will generate a lot of spikes because they don't have enough, probably not enough of this local inhibition, but we're also measuring excited inhibitory currents in those cells. We also look whether these effects are local or distal. Those are my last 3 slides. By using a PAX5 Cre mouse. So in this PAX5 Cre mouse, we can inject a Cre-dependent virus that then becomes fluorescent upon mixing with a Cre. So we inject it in the VTA and in SN. And what we could see is that these, so here in SN, we saw that the PAX5 positive cells make putative synapses on the TH cells. And now we have to do super resolution to really see that this is a synapse, because right now you can only see that this is a putative synapse. But what is also very cool is that it's not just interneurons that are affected. So the PAX5 is not just important for the inhibitory interneurons that are within SN and midbrain and VTA, but it's actually, if we now traced these neurons, we saw that there's an SN to the thalamus that are PAX5 positive, SN to caudate and putamen and striatum. And then when we looked at VTA, we also saw axons in the amygdala and the nucleus accumbens, meaning that these mice, and then by extension, probably the patient, is not only lacking inhibition in the VTA or SN alone, it's actually lacking this wide range inhibition onto nucleus accumbens, onto amygdala. So this is something that we are pursuing right now. This is just an overview. So in general, when we think of both the TSC1 and the PAX2 and PAX5, we are very interested in the lab in this quantification of the inhibition excitation disbalance, particularly during development in the midbrain, and how it can lead to hypo or hyper sociability and predict this behavioral changes. So with that, I would like to, before I thank you for the attention, I should say it really takes a village to do a project like that, particularly with patients. I am not an MD. I cannot really, draw blood or anything like that from a patient. So I am very privileged to have a lot of collaborators and a fantastic lab, and also some institutions who believe in this work and support us financially. So thanks a lot for your attention.
Thank you very much for this fantastic, fascinating lecture. Okay, we have time for questions and comments. Not yet, so maybe to warm up the floor, I will start. You mentioned about critical periods. and visual work on visual system. Let me say to youngsters in the audience that in the brain development, critical periods are really, really important. If you miss them, you may simply lead to defects. One of the most known is critical period for speech learning, right, which involves cerebellum as well. We know the cases when child miss the critical periods when they haven't learned and they were unable to learn speaking, talking in the later on. Now I'm recalling Takao Hen's work from Harvard back in the day and others when they others especially when they study birds singing in critical periods, right? And the study leads to the discovery that application of benzodiazepines can reopen critical periods, allowing for plasticity, which leads to at least partial learning after missing critical period. Can you comment on this? Could be this also part of therapeutic approach in various brain disorders, including autism?
Very fascinating and multifaceted question. So I think I will first start is that these critical periods for song development, they are actually cerebellum is very understudied for the song development because that's just really sequences of the muscles, whereas there's the L-man and the other nuclei in the bird. Birds have like, if we have, mammals have like a brain like in layers. They are, birds have basically just like nuclei that are together, but you can find analogous structures. But they do have also quite a big cerebellum. So anyway, they found that these are some of those nuclei that are very important, and they really have a very, very strict critical period. For humans, for speech, I think most of the people nowadays studying speech think that there's more of a sensitive rather than critical period, meaning that those kids who have never learned speaking, they were also deprived of other input. But if you think of learning a second language or third language, etc., that it's probably more of a sensitive period rather than critical. So there is still some plasticity left over, whether in some of the songbirds, it's really like, you snooze, you lose, you don't learn a song, you're never gonna sing. So that's one difference. The second thing is actually in the cerebellar work, when it comes to critical periods and sensory motor learning, people think now of perineuronal nets. So rather than benzodiazepines, there's actually work in reopening critical periods by loosening perineuronal nets. So neurons are very tightly packed. There's a lot of astrocytes, glia cells, etc. And this is all in this 5 fibers that are called perineuronal net, and they stiffen with age. And this is something that if you want to grow a new axonal collateral, or if you want to grow more spines, maybe you need to loosen this perineuronal net. So this is definitely, this is actually a project that is ongoing in Amsterdam to see about this loosening of the perineuronal nets. For benzodiazepines, I haven't heard of any ongoing study. It would be interesting to look at because they don't have a lot of side effects. And they're, of course, already FDA and EMA approved. So those would be the drugs. I'm not familiar with any of these, of any ongoing studies on that, but yeah, I'll have to look into it. But that would be interesting point of possible intervention.
Thank you. And who from Amsterdam is working on this perineuronal net? Do you remember? Okay, maybe I'll later to write down because my memory is so bad these days.
Yes, I will write it down.
Thank you. Other question, comments? Vitek.
I have a question about TH neurons. So can you distinguish that what you see is not the intrinsic excitability rather than the effects coming from the synapses and from the inhibition?
Very important question. This is where we're also recording the mini excited inhibitory currents. And so this was just the first study, and we also see that there's less minis. So this is why we can see it, but I don't have the full data set yet, but we're doing it right now, but it's very important. So we do these because you do this with different blockers, right? In order to record minis, then you block the spiking to only record the currents. Then we did not do it all in one experiment. And we just got some funding. Actually, I should add to the slide because a PhD student is going to start next week to continue on this project. And she's also going to do in vivo recordings in an awake animal where there's the whole network. Because of course, in a slice, you cut off a lot of inputs. We do horizontal slicing for Midbrain, but you still cut a lot of inputs. So you only have the local inputs, both excitatory and inhibitory. But we are really interested in doing the recordings. And I was playing, but we only do electrophysiological recordings, but the lab of Ingo Bilun in Amsterdam is doing really great work with also measuring dopamine release. So this is something that I hope the PhD student was going to establish this collaboration, but then this we have to start up because we would also like to see whether there's more dopamine being released in the striatum in those mice, and that would
because such excitability would explain your hyperactive phenotype as well, right?
Yes,
because
exactly.
It's quite huge. And I understand that in this model, the effects of PAX2 mutation are developmental. What would you expect? What would happen if you delete the PAX2 in adulthood?
This is PAX5, but yes.
PAX5, sorry.
Yeah, actually. PAX2 completely, there's no more expression in the adult. Here in the PAX5, we still have some, but it's very, very little. So we only see it in some cells still in the midbrain. We do not know, so we don't have this mouse model. We tried to make it, and then the mouse model failed, but we're going to probably try to make it again, to make the tamoxifen inducible.
In PV cells, this mutation. You would like to target the mutation to PV.
Exactly. So the mouse was supposed to be done last year, but it's not yet. But we will be able to answer it if we have tamoxifen dependent is when you make a mouse mutant, but then you can delete the gene not from the beginning, but then you give tamoxifen to a mouse and then you knock out the gene. So the tamoxifen-dependent mouse is something that we're going to look at. But I don't expect huge side effects. I do think that are mostly... So definitely the loss of the interneurons and loss of the inhibition is developmental. We think it's a lineage determination. We know that transcription factor PAXs in general, they determine at the moment where the cell divides and then it proliferates and then it gets sorted to either a GABA cell or a glutamate cell. This lineage defining model is when these transcription factors activate relevant genes. The big question that we're trying to answer now with Jeroen Pasterkamp is, are these inhibitory cells just never born, or are they born and they die? We don't know that yet.
Okay, and the last technical question about the experiment in the beginning that you showed with rats.
Yes.
You showed two groups, the one that was treated with CNO and one untreated.
Yes.
Do you think that the effects that you observe may come from the conversion of CNO to clozapine?
We always, I think I showed that, I'm not on a slide, but I have it and it's definitely in the paper. We also always have non-dreaded CNO group. So we have just a vehicle group, a CNO group, a dread with a vehicle, and then a dread with CNO. So we didn't show all these graphs, but in the paper we always have all four groups. And actually nowadays the standard, because that we started doing it in 2015 to publish in 2018. And now the standard is actually have another group that's GFP positive, just to make sure that expression alone of something in a cell is not causing a problem, just like with the opto. And this is actually because of the optogenetics that showed that particularly if you express an opsin that inhibits that can be actually leak chloride just by its own, changing the activity of the cells. So when you do optogenetic studies, that some of these can actually be already, without the light alone, can already have effects. But we had the 4 groups.
Thank you very much. Time for last question, comments. If not, maybe me, last comment. So you mentioned about the German guy who came to you with weird mouse. I want to make a comment that in case you're interested, I have very weird cat at home. It's really weird. He doesn't like me from some reasons. But seriously, maybe some of our house animals may also have special needs. So this is something to be aware and mindful about. I believe that our youngsters in the audience may be a little bit too shy to ask questions right now in public, but to remind you, we have in a moment a session when young scientists can freely talk with our speaker. Let's thank Aleksandra for this wonderful talk. Thank you very much.